Last night I uploaded few of the videos of the topic synapse, and as I said, while conversion of those narrated slides into videos, somehow certain parts were missing. And out of those certain parts, the important one of them is inhibition at synapse. And inhibition at synapse, there are four types of the inhibition at synapse. This one I will try to cover it. One is the postsynaptic inhibition. Second one is presynaptic, feed forward or feed backward. It means, like, say, if we take the example, this is the presynaptic neuron and this is postsynaptic neuron. So either the inhibition can happen over here. This neuron gets excited, stimulated, releases the neurotransmitter, and these neurotransmitter makes the postsynaptic neuron inhibitory. Or what does it mean? Inhibition means it will go into hyperpolarization. So that thing is called as postsynaptic inhibition. The second one is presynaptic inhibition. So presynaptic inhibition means we are taking this example of the presynaptic neuron, then this is the postsynaptic neuron. It gets excited, this neuron will get excited, will release the neurotransmitter, and if in turn, say we are providing one stimulus over here, okay, and that stimulus is supposed to release like 100% quanta of neurotransmitter, but instead of the 100% of the neurotransmitter, if it is releasing say 80%, what can be the reason? Now we are stimulating this neuron with the same strength of the stimulus, but still it is releasing lesser amount of the neurotransmitter. It means there is somewhere over here there is one connection with the other neuron which is making this neuron to release less neurotransmitters, and all these are occurring before the synapse. So that's why it is called as presynaptic inhibition. Okay, then there is one more term, that is a feedback inhibition. Means, say this is the presynaptic neuron and then this is the postsynaptic neuron. What does this presynaptic neuron do? It makes it excited. And what is this postsynaptic neuron is supposed to do? It is supposed to excite or inhibit the next one, depending on whether the neurotransmitter released is the excitatory over here or the inhibitory. Just imagine this is excitatory neuron, okay, and this one is inhibitory. When we provide a stimulus to this particular presynaptic neuron, it will release the excitatory neurotransmitter, and excitatory neurotransmitter will depolarize this postsynaptic neuron. Upon depolarization, it is supposed to release the neurotransmitter. And what kind of neurotransmitter it will release? Inhibitory, right? So if this postsynaptic neuron has got one branch that is having connection with the presynaptic neuron, then what will happen? We provide the stimulus to this neuron number 1. Neuron number 1 will release excitatory neurotransmitter, and these excitatory neurotransmitter will make this neuron number 2 to get depolarized. Okay, and when this neuron number 2 gets depolarized, obviously it will release the neurotransmitter at its branches. One branch is over here and the second branch is making a synapse with this neuron. And what kind of neuron it is? It is inhibitory. So it will release inhibitory neurotransmitter over here. So because of the stimulation of the neuron number 1 by that stimulus, it is trying to make this neuron to release excitatory neurotransmitter, but it is also getting one branch from the postsynaptic neuron which is inhibitory. So there will be less amount of excitatory neurotransmitter release. This is called as feed backward. Okay, feed backward. So in the coming slide, one by one, we will cover all these things. Synaptic inhibition, we said there is generation of the IPSP, but in this one there is happening of inhibition, but IPSP is not produced. Okay, the C1 is inhibited A neuron. So the reason is, just consider the example of C. This was the C neuron and this one was the A neuron. A has got connection with the B and C has got connection with the A. So we will talk about this thing, like inhibition occurred over the A. And what is the mechanism of the inhibition? The mechanism is either whatever the neurotransmitter is getting released from the C, because it is inhibited, it will release either the glycine or the GABA, right? So this glycine or the GABA, it will get attached to the receptor over A and create some sort of the, like some reaction will happen. By reaction I mean to say, like it will get attached to the receptor and it will make it lesser depolarized, or we can say it will make it hyperpolarized. So what is the mechanism? One of the mechanism is by opening of the chloride channel. Like the inhibiting neuron C releases an inhibiting neurotransmitter which binds to GABA-gated chloride channel. Like say GABA is getting released, it will get attached to the GABA-gated chloride channels, and through the opening of this chloride channel there will be more and more negativity inside of the neuron. So if there is more and more negativity, it means it is moving toward the depolarized state or the hyperpolarized state. It will move toward the hyperpolarized state. That is one of the mechanism. The second mechanism is this neurotransmitter, inhibited the GABA neurotransmitter, it gets attached to G protein. Like say, see, the GABA is getting released. It can have many receptors. One of that receptor can be GABA-gated chloride channel, and the second one can be G protein. So when this GABA gets attached to the G protein, bind to a receptor that is called as GABA receptor G protein, which in turn will activate this G protein. Say the thing is, like if you read about receptors in the endocrine part, say this is the GABA receptor. GABA is getting released from the C neuron and this GABA receptor is at the surface of neuron A. This is A neuron surface. Now after the attachment of the GABA over this receptor, there is G protein, like say by this, consider this is G protein. Now this G protein will get activated. Okay, and with this G protein after activation, it will help in reducing the amount of excitatory neurotransmitter release from the neuron A. See, this was the neuron A. It was supposed to release excitatory neurotransmitter over the neuron B, but in the way C released GABA and that GABA got attached to the G protein GABA receptor, and which activated G protein will make this neuron to release less amount of the excitatory neurotransmitter. How it will make it to release less amount of the excitatory neurotransmitter? One of them is by opening of the potassium channels, and second is by directly blocking the calcium channel. If it makes the opening of the, say, I'll try to do this thing, like say this one is A and this was C, GABA attached, and it can reduce the excitatory neurotransmitter by these two things. If we recall back, say this is the synaptic terminal of A and neurotransmitter are getting released. How they are getting released? There are the calcium channels, and through these channels, when the wave of depolarization reaches over here, it will open up the calcium channel. Calcium will move inside, will get attached, bind to the vesicles which are carrying the neurotransmitter, and these vesicles will get attached and exocytosis will happen. Okay, but if these calcium channels get blocked, then there will be less entry of the calcium, less release of the neurotransmitter. That is one mechanism. Second one is by opening of the potassium channels. Say if there is opening of the potassium channel, the potassium concentration is more in the ICF or the ECF. It is always more in the ICF. So as soon as potassium channel gets opened up, potassium will try to move along the concentration gradient, which means it will move outward. When the potassium moves outward, the positive charged ion moves outward, the inner will become more and more negative. If it is becoming more and more negative, what does it depict? It depicts that it is getting hyperpolarized. So lesser depolarization, less release of the neurotransmitter. So these are the two ways, by opening of the potassium channel or by directly blocking the calcium channels, by which the GABA act through the GABA receptor, G protein attached GABA receptor. Okay. So it's